guys, welcome. If you are joining me, I am just setting up. If you are joining me on YouTube, welcome. If you are joining me on TikTok, then welcome as well. For some reason, it is not working the way I wanted it to work tonight. Literally trying new things today. Trying different things in different ways to see how it goes. All about practice, all about trying things, all about experiencing what is going on. So if you are with me on YouTube, welcome. Please do chat away. If you are with me on TikTok, then please ask away as well. I am just setting up and figuring things out, so we'll just see how that goes. Let me see if I can get this to work the way I want it to. For some reason, it doesn't want me to do it on the question. 77, what do you want, past paper? Which one would you want to look at? You got a specific paper, specific question that you're struggling with? Which one are you wanting to do? GCSE combines. So at 77, I'm actually doing A-level now. So my GCSE I did earlier today at 6:30. So now I am doing A-level, answering some A-level questions. So if you want to have a look at my videos, I've got some playlists on my channel which are GCSE AQA specific. So if you want to check them out, please do check them out. I'm just setting this up. It's not working the same way it did earlier. I'm trialing new things, but yeah, this is more for A-level students now. I will be live again on Monday next week for the last live I'll do before Christmas for GCSE. So if you do have specific questions, then please do ask away. Gel electrophoresis and PCR cycles, of course. So Susie, I can cover that. Now for some reason I can't get it to work on here like it is on my channel. So if you want to join me on my channel, you'll be able to see my board, and I'll draw and go through what's going on there for you. So please do pop across and join me. But with regards to PCR and electrophoresis, the one thing to note whenever you're doing the PCR and electrophoresis is understanding DNA, and the structure of DNA is the first component to get you to where you need to be for understanding what's going on with your electrophoresis. So give me 1 second, I'll copy this across and I will go through this on here. So if we are going to extract DNA, okay, so Omar's just asked a question with regards to what's the difference between an antigen and an antibody. So that's really important to note the difference between the antigen is the one that's on the surface of the cell, which distinguishes whether it's a self cell or whether it is a foreign cell. An antibody is what your B plasma cells produce as a protein that's going to bind to the antigen. It will be specific for the antigen, and what it will do is it will bind and either neutralize, or if it doesn't neutralize, it will agglutine, clump them together, or it will do opsonization. So it's a case of what are you looking for in the definition between the two. One's on the surface, whereas one will bind to the other. Oh, thank you. Adele's been watching all my videos. Thank you. I hope that you find them helpful. Right, I'm going to do a little bit on PCR and gel electrophoresis, but I will try and answer questions as we go. So if you do have questions, if someone, whoever's watching on YouTube, if you could write in the chat just so I can see that it is actually bringing up comments there. If you have any questions, please do ask away, and I will try. Yeah, Susie, if you jump over there, you'll be able to see my actual board and what I'm going to draw and write and the animations that I've got. Oh, perfect, Silly. Amazing. Jump over. Come and see me. And definitely make a thing of putting something in the chat so I can see whether it's working. I'm trialing new things right now and I'm hoping you can hear me on YouTube with this. So yeah, do let me know if you can hear me, if you can't. Right, so what we're going to do is we're going to extract... Yes, hi, Silly. Thank you for joining me on YouTube. Can you hear me okay? I don't know why your name's weird either, but it's nice to see you, whether it's Silly or whether it's MQW9EP. Thank you for joining me. Okay, so what have we got? If we're going to look at PCR, polymerase chain reaction, what we're going to do is we're going to amplify a sample of DNA. So we need to extract the sample of DNA from the cells. Now, we know that DNA is a double helix. We know that it has millions of bases. So what we need to understand, you're very welcome. Susie, if that's you, no name. DS1NG. Hi. What you have to understand is these restriction endonucleases, right? What do restriction endonucleases do? Well, what they do is they're going to digest the DNA strand into varying lengths. So you know that DNA has got a sequence of the bases. So please do ask questions as we go, because I want to help you understand. Now, I'm just going to make this up at the top of my head. I'm not giving you the actual sequence. There's some teachers out there who do know this stuff off the top of their head. My brain's so filled with all the other information, I'm not going to worry about that. I should learn it, but I'm not going to. So I'm just going to imagine that it's... Oh, crumbs. Let's try that again. That it goes A, T, T... A, C, G, let's say, right? So there is my sequence, and then the straight line is representing all the other sequences. But this is the only one I'm interested in, is the ATTAGC. So what we're going to look at is what's my complementary base pair. So A pairs with T, T pairs with A with T, C with G, G with C. So basically what we're looking at right now is we're looking at specific recognition sites, and these recognition sites are recognition sites for a restriction endonuclease. So an endonuclease, A's, means it's an enzyme that's going to cut endo in the inside. So we've got a restriction endonuclease that's going to look for, oh, thank you. It's going to look for a recognition site. And I'm going to say it's this, I've just made it up. And what's going to happen is when it locates the recognition site, it's going to cut at those bases. Now, what do we need to remember? Going back to our previous knowledge, we know that we have got a nucleotide, and the nucleotide is attached to another nucleotide by a phosphodiester bond. So what we have to do is the restriction endonuclease is going to cut the phosphodiester bond between the A and the T and the G and the C at the other side. And so what we therefore need to understand is when we have these restriction endonucleases, for example, I'm going to take one which is called EcoRI. You do not need to know these names, just to be clear. So EcoRI, if we take the restriction endonuclease from EcoRI, what's going to happen is it's going to cut wherever it finds the recognition site that it's looking for, okay? So whenever it finds that recognition site, it's going to cut at that position. So in this case, I've represented it with my purple lines here. So we've got purple line, purple line. So what will happen is, because it will cut at that recognition site, I get a really small fragment, I get a longer fragment, and then I get a fragment that's bigger than the original one, but smaller than here. Okay, so at any place along all the DNA sequence, wherever there is a recognition site, it's going to cut at that specific point. So it's looking for that combination. And we use ones that we know of, just to be clear. And as I said, I should know them, but I don't. I got to use a restriction nuclease with a micropipette in my work experience. Oh, amazing. So I've actually done this with my students before. So we were very lucky in that I have connections with Cambridge University. When I did my PGCE there, my lecturer, Mark Winterbottom, amazing lecturer. He was so energetic, so full of joy. He was very much about having the practical side of biology, and so I've tried to ensure that I kept that practical side going on. And so I got the opportunity to actually bring Amgen Bruce project to my school that I was working at. And so Amgen Bruce is a company in America that donates their electrophoresis kits and very small samples of DNA, and we actually transformed some bacterial plasmids, and I'll show you them in a minute. But what we did as one of them is we took a swab of our cheek cells, and then we put them into a sample tube with the restriction engine nucleases, and it was cutting for specific recognition sites. So NXL-A, to be honest, I haven't looked at NXL-A's questions in a long time. I taught NXL-B last year, but again, it's about making sure the specification along with the questions that are being used are specific to you. So I don't know them off the top of my head, but I do know the NXL-B ones because I tutored last year with that one. But I actually really like the NXL questions. They're quite different, but content-wise, biology content is pretty similar across all the exam boards, apart from there'll be different emphasis on certain things. So AQA and OCR are so similar, 75% overlap, but I have to be looking at that specification when I'm teaching it to make sure that I'm teaching the right stuff. Habib, I did see your message on TikTok as well. I am trying to keep on to. I'm going to do this and then I will go into that. But if you go to my patterns of inheritance videos, Habib, you'll find an epigenetics one in there. It's either labeled as epigenetics or it's labeled as, no, I think it is labeled as epigenetics in that playlist. It's definitely there. I'm currently, interestingly enough, I'm at the epigenetics part of my patterns of inheritance workbook. Study guide, it's almost finished. I'm super excited, really sad, super excited though, because I've been putting so much effort into this and I've been making like little diagrams. Today was about mice. But anyway, back to this. So restriction engine nucleases, we've got enzymes that are going to cut at specific points. When they cut those recognition sites, depending on where those recognition sites, if I use the same DNA strand in this example, the first one, EcoRI, cut in 2 places. BAM H1 is only going to cut in 1 place. So that's going to leave me with 2 fragments. And if I were to put both of those restriction engine nucleases in together, they will cut at the recognition site. And what will happen is I'll end up with 4 bands. So we use restriction engine nucleases that we know because we know they're going to digest at specific recognition sites. And therefore we know we're going to end up with bands. And it's about how many bands, where they separate into, so that we can get a picture of the DNA sample that we've taken from, whether it's a crime scene, whether it is a paternity test, whatever that is that we're looking at. We're going to start with these restriction engine nucleases to cut up our fragments. Now, the next thing we need to do is the polymerase chain reaction. So you just have to bear with me as I move some stuff over so you can see. So what is a polymerase chain reaction? A polymerase chain reaction is where you're going to do exactly as it says. You're going to use polymerase. Again, it's about biology and the words. Polymerase is an enzyme. Well, you've already heard about the RNA polymerase, DNA polymerase. So therefore, in this case, we're going to use an enzyme polymerase, which is going to be used to copy. Well, what do we know about copying? We know when we copy, we're going to do DNA replication. And we know that DNA replication is semi-conservative. And so what we're going to do is we're going to use the knowledge of semi-conservative replication. Yeah, Lou, it's not working on here. For some reason, it didn't work the same way. So if you jump over to you need to go to my YouTube channel. I don't know what's going on tonight. I'm trying new things and it works slightly different. So you need to jump over here. So the first thing that's going to happen is we need to denature the DNA strand. So normally when we do DNA replication, we are going to break the hydrogen bonds by using an enzyme, helicase. Well, we're not going to do that with polymerase chain reaction because we want to limit the number of different molecules and contaminants that could be there. So therefore we want this to be really controlled. So because we want it to be really controlled, we therefore need to put it to work basically in only the places that we need it to work. So give me 1 second. Sorry guys, give me 1 second. I just have a video I need to edit from my group tuition that I did earlier before tonight's session. Right, so because we're not going to put helicase in there, instead we need a way of breaking those hydrogen bonds so that we get our template strand. So using all our knowledge that we had from DNA replication, the fact that it's semi-conservative, the fact that you need to have your template strands, then what we're going to do is we're going to heat it up to 96 degrees C. So in this little machine, which is called a PCR machine, what's going to happen is that is going to heat to 97 degrees C. That will denature the strand and therefore it's going to break the hydrogen bonds between the base pairs. Yes, be careful. Susie, be careful. The antisense and sense strand we only tend to talk about when we're looking at the transcription side of things, not when we're doing DNA replication. Because DNA replication, we are going to be copying both strands, whereas transcription, we are going to have our mRNA make a copy of the antisense strand, so therefore it's the same as the sense strand. So we only tend to talk about the sense and the antisense strand when we're looking at it on transcription translation. Why do the phosphodiester bonds not break? That's a really good question. The reason the phosphodiester bonds don't break is because, remember, a phosphodiester bond is a very specific strong covalent bond. It's a covalent bond between the phosphate and the two esters at either side. So when we are heating it up to 96 degrees C, we are breaking the weak intermolecular forces, which are the hydrogen bonds. So you've got to remember the difference between a covalent bond and an intermolecular force. So in this case, when we're denaturing it by heating it to 96 degrees, that will get it to the point where we can break those hydrogen bonds between the DNA bases, but not break the phosphodiester bonds and not damage the strand. So I'm covering this, Habib, on polymerase chain reaction, which is for both AQA and OCR, but I'm going to make sure that I try and cover each bit as much as I can. I don't think there's actually anything different, to be honest. I just haven't done AQAs in a while to double check it. Right, so what's the next thing we're going to do? Well, the next thing we're going to do is we're actually going to drop the temperature down to 50 degrees C. So first of all, we had it up at 96 degrees C. Obviously, increase the temperature, increase the kinetic energy, therefore allowing for those hydrogen bonds to be broken. We're going to drop it down because we want to not cause a problem, and we need it to do the process of reforming hydrogen bonds. So we've broken the bonds between the two strands, but now we want to form bonds, and that's going to be used through a primer. So this DNA primer, so you can see here I've used a 5 nucleotide chain. So this DNA primer, which is a short sequence of DNA bases, which is complementary to a known part of the strand. Okay, it's really important that we understand it's a complementary part. So what we're going to have is it's going to come along and form hydrogen bonds. And the other thing to remember, now this is OCR, not AQA. Remember, you have to know the difference between a purine and a pyrimidine. So always a purine and a pyrimidine will come together forming complementary base pairs. So for those of you who know you've got your thymine and your cytosine, your thymine is a pyrimidine, your cytosine is a pyrimidine. A pyrimidine is a single ring. Oh my gosh. The one thing I would say to you, if you're anything like me, my brain works so quick that sometimes my hand can't keep up. And so therefore I forget to write letters, or I have to double check. Oh, interesting. I like that. Pyramid, 1 peak, so 1 ring. That's how you remember it. That's the thing, is find what works for you. I do it where I use the alphabet. I've always used the alphabet because for me, words are quite difficult for some reason. So I know that a double ring is a purine because of the letter U. And so therefore, that means my adenine and my guanine have to be purines, because my thymine and my cytosine both have Y's in them, so they're pyrimidines. That's how I remember it. So we're going to have this complementary base pairs happening where the short sequence of DNA is going to complement that single strand template, and it's going to bind. And so therefore, the reason that has to happen is because DNA polymerase cannot build from nothing. It needs a primer to start from. And it's interesting because we don't go into that detail at OCR or AQA, but even during replication, it actually does need a primer. Anyway, let's move on. So we've dropped the temperature down. We've dropped it down to the point where we can actually have it doing the process called annealing. Oh, crumbs. Do not do that, Christine, because that will move everything around. So there we go. We've got a primer. Our primer is going to come along and it's going to form these complementary base pairs between your purine and your pyrimidine. And then what's going to happen is we're going to move the temperature back up again. So we're going to raise the temperature up to 60 degrees C. When we raise that temperature up to 60 degrees C, what's going to happen is the Taq polymerase. So we're now using a very specific polymerase, one that can withstand higher temperatures. And what's going to happen is it's going to come along and it's going to make its way. And as it's making its way, it's going to build the phosphodiester bonds on this strand. Now you can see I've got 3 nucleotides. So the key thing to remember is I've also had to give 3 nucleotides. And those nucleotides are going to form complementary base pairs. Yes, this is year 13, and you will go into more depth on it, Habibi, if you are currently year 12, and I think you're AQA, so therefore this would be, I want to say it's module 6 for you, but I need to double check on your spec. It's definitely module 6 for OCR. Okay, so what is polymerase chain reaction then? So polymerase chain reaction, we've got original DNA strand. We know that we need 3 nucleotides. We need a primer. We need our Taq DNA polymerase. And the machine is going to go through cycles of denaturation, annealing, extending. So it's going to go temperature down, module 8, thank you. So module 8, Silly's helping us out here. This is what I need for you guys to help with each other in knowing whether it's which module. OCR, I can tell you without even thinking about it, but with the AQA, I have to double check, because although I do tutor a lot of AQA students, I spent a whole load of time building these resources for OCR, and so therefore it is stuck in my head for the OCR. I will at some point build the AQA. I keep saying that to my students. I will adapt it for you. But yeah, at some point, once I've got all the resources and they're a good quality. Anyway, back to here. One DNA strand. The first time it happens, what's going to happen is that DNA strand is going to be separated, and we're going to have a new molecule which is made up of one template strand, original strand, and one new strand. So therefore, because of semi-conservative replication, you end up with 2 copies of the original strand. And as I keep going and keep going and keep going and keep going all the way to 30 cycles. So when I did this with my students, it took 2 hours. So as I said before, we took a swab inside our cheek. We then put the cotton, I don't even know what to call it. It was like basically a cotton tip that you would not stick in your ear, because don't ever do that. But it was the same sort of thing. You take a sample, you placed it into the jar, and then you had to really rub it around so that you were dislodging as many cells that you managed to get into the primer, the nucleotides with the DNA Taq polymerase. And then we had to put it into the machine and leave it for 2 hours. And what that does is that amplifies the DNA that you had. So a small amount is now being amplified to almost 2 billion copies of it. So to work out your final number of the copies, you take the initial number, which is 1. So I have 1, then I've got times 2 to the number of cycles. So 2 to the 1 cycle, therefore 2, and so forth, 2 to the 2. And if I start with 10 copies, I would then do 10 times 2 to the 30 cycles. Because it's 2 to the power of 3. Oh, no, that's right. Yeah, you're right. I made a mistake on my slide. Thank you. Should be 2 to the power of 4. That's exactly what it should be. No, not your bad. Thank you for picking me up on that. Because that's the thing of sometimes, even us as teachers, we'll make a mistake because we're thinking something and then we write, and I do that all the time, my numbers. So do make sure you double check your answers too. Because if you don't double check your answers, that silly little mistake that I made there could have cost me the difference between getting an A star to getting an A. And that's okay, in the sense that I know that I make these mistakes. I'm comfortable with the fact that I make these mistakes because I learn from making these mistakes and I double check. And when I'm trying to do things really quickly, if I try and throw them out too quickly, then these mistakes will happen. And in the lesson, I will actually try and catch my students out by doing things like this on purpose. Sometimes it's not, and I'll pretend it's on purpose, but majority of the time I will be trying to catch you out. So don't ever apologize for asking questions and double checking things that your teachers have put out there, as long as you do it in a respectful way. That's the one thing I would say. But no, we are human. We will make errors. And it's about how we deal with them and what we learn from them. And that's what you have to realize as well. You will make errors, but you learn by putting it down on paper. If you don't put it down on paper, you think you know what it is that you have as knowledge, but the minute you put it down on paper, it's maybe not going to go into the right depth, or it's maybe not going to be correct, and you start to figure out those misconceptions. So don't apologize. I'm glad that you're here and asking and checking, because that makes me double check my job and it makes me better at what I want to do, and that's to help you guys. But also be comfortable being uncomfortable. I've been doing this for over 20 years and I'm always uncomfortable, so I'm like, yeah, that's fine. We're all good. We made a mistake. But working this out, 10 times 20 to the 30 cycles. Therefore, I get this really large number. Now, remember we used restriction endonucleases before. So basically, if I take this, see if I can do this, she says, actually no, give me 1 second. What I will do is I'll just do it really quickly. I will draw it really quickly. So we've got strand, strand, strand. Because remember, I used restriction endonucleases. So therefore, because I used restriction endonucleases, I had different length strands. So the replication is going to happen on each of the strands, which have different fragments. Okay, so that's another thing to note, that they're going to have different strands because they're going to have different lengths, because we use restriction endonucleases before doing the polymerase chain reaction. So what do we do then? Well, we do what's known as gel electrophoresis. So we're going to take some proteins, potentially, or we're going to look at DNA fragments. So because we're looking at polymerase chain reaction, it would be the DNA fragments, but this can also be done on proteins. So students get hung up with gel electrophoresis and DNA and PCRs, and then they get asked the question on proteins and they're like, but I only knew it happened with DNA. And it's like everything they learned went out the window. It doesn't go out the window. It's just about applying the knowledge that you have to whatever they're asking the question on. So proteins and DNA fragments can be separated by gel electrophoresis. So what does gel electrophoresis actually do? Well, what we do is in here, you've got these wells. So basically you have an agarose gel. So it is a gelatin block. And what they do is they put a comb in it. So depending on how many wells you want, depends on the size of the comb. But anyone who has done this basically gets this comb, and then what they do is they make the gel a little bit bigger than the comb, and then once it's set, they pull the comb out. So basically what you end up with is, if I remove my comb, let's try that again, what will happen is it will actually leave an area where the comb was positioned. So in the lab, they had like a bin full of used agarose gel. It looks so, it is disgusting, but it's basically just gelatin, which is used to have a material for the DNA to move through. So it's I like to think of it as a bit of a weird one, but I like to think of agarose gel when it comes to gel electrophoresis a bit like an assault course, where you've got on the ground, it's going to sound so bizarre, but it works for me visualizing it. On the ground, what you have is, you know, like the netting. If you've ever done like a Tough Mudder or an assault course, anything like that, and they've got like this net on the ground, you've got to crawl under the net and you've got to make your way through the net and get from one side to another. Well, I always like to think of agarose gel a bit like that. It's this basically this jelly net mesh thing where you have to figure your way through the net from one side to another. Okay. Now, if you are really small and petite, you're going to be able to make your way through quite quickly. But imagine you're not small and petite, you are wrapped up, you have a rucksack on your back, or you have a rucksack on your back and your front, and it's going to make it much harder for you to get through. And so therefore, what's going to happen is, if we put an electric current through a trough, now it's important to remember that we need the electrical current to flow. So because we need the electrical current to flow, we need there to be a buffer solution. A buffer solution which is going to maintain the pH, but it's a solution that allows the electrical current to flow. So we have the cathode, which is negatively charged, and we have the anode, which is positively charged. This is where GCSE chemistry comes into your A level. You should know that DNA is negatively charged. And because it's negatively charged, if I put an electrical current through the electrophoresis kit, what's going to happen is the negatively charged DNA is going to be repelled from the cathode and is going to be attracted to the anode. Now, like I said before, these fragments, a bit like you going through the net, these fragments are going to move at different rates. And because they're going to move at different rates, what's going to happen is the smaller fragments are going to move through faster than the larger fragments, which are going to move through slower. And so what happens is they separate out into these bands. But what's missing from my diagram is I should have another well that gives me a DNA ladder. A ladder which is of known DNA bases. So whenever you do electrophoresis, you normally will use a DNA ladder so you can determine, well, whatever size this one is, that must be that size. Whatever size this one is, that must be that size. So you use that as a way of determining the size of the DNA fragment that you have in your sample. Now, DNA will then separate into these bands, and they'll separate according to the size. They won't separate out like this, because it could be that actually somebody has all the strands in their one well because the restriction endonuclease is separated them in that way. So if I come back to here, this one here, so remember we talked about these restriction endonucleases and the fact that they're going to digest the fragments down, the fact that they're going to separate them into bands using electrophoresis means, if I just make that a bit smaller, there we go. What we've got is, if that's 10 kilobytes of base pair, kilobases even, 10 kilobases, 8 kilobases, 6 kilobases, 4 kilobases, 2 kilobases. So if I use the EcoRI, I've got my 3 fragments, one that's really small at 2, one at 4, one at 10. So I can conclude that this was 16 kilobases long. Is this quantitative or qualitative? But still you're comparing it to see how. So it's a bit of both. So it's quantitative if you've got numbers and you're using a DNA ladder. However, qualitative because it's observational. You're seeing the band, but you could potentially measure the width of the band as well. And if you're measuring the width of the band, that will help you to get quantitative data. So it really depends what it is you're actually getting as to whether it's quantitative or qualitative. So just visualizing it, it's quantitative. By using the ladder, we're making it quantitative. And that's really useful when you're doing genetic engineering. If you're transforming a bacterial plasmid, you can use that as a way of confirming whether the plasmid has taken up the gene that you desire. Okay, so obviously we talked about this one, and this one is actually separating into 2 strands. Now, if you notice, this is a thicker band, and that's a thicker band because we've got 2 lots of the 8 kilobases. So they're going to stop at the same point, but because there's 2 lots of them there, it's going to make the band a bit thicker. And if we look at this one, obviously, we're separating it into 3. We're going to get our different sizes, but we can determine 2 8s, obviously it gives us 16 kilobases. On this one, what have we got? We've still got our 2, we've still got our 4, but now what we've actually done is we've broken it down to another 4, and this one's a 6. Oh, amazing. Yeah, we did something very similar, but it wasn't Mycobacterium. I'm not entirely sure which, I should be able to look back and see, but I can't remember which one we used, but yeah, it's so cool when you actually get the opportunity to do this in real life situation and to be able to see what it looks like. So this is actually one of the plates that we used as a way of visualizing. So this is the ladder. It's got a bit of a blur to it, but this is the ladder that you can see here. And then we were looking at whether or not they'd managed to, one, actually cut it up. So we used the restriction endonucleases as one sample. One where we'd added in the enzymes to join them together, so ligase. And one where we had not put the ligase in and we were looking at what happened. So it's really cool when you get that opportunity. No, Mohammed, the curriculum hasn't changed from last year. So hybridization, what is hybridization? So hybridization is where the gel that you have put through gel electrophoresis actually gets immersed in an alkaline solution. Oh, it's so hard. It's so hard to put it in. It's so funny as well, because what you tend to do when you do it, if you've got your micropipette, it's always one of those things where you need something to lean against. So you're always told to use your other arm because it's actually really hard. And you see all these students shaking and sticking the micropipette into the gel, and it's a really tough skill to learn. And I've never had the opportunity, I mean, I've used one to play with, but I've never had the opportunity to do it in the lab. But seeing them where they have like about 10 different needles all at the same time that you can put it in to each of them simultaneously just makes it so much easier. Whereas we were doing it one after one after one, which is actually quite tough. But yeah, so when you actually use this alkaline solution, what that's going to do, obviously we're changing the pH, we're going to denature the DNA strands, and therefore we're going to actually make them single-stranded DNA fragments. So we use this alkaline solution and we remove it, and so we can then do what's known as southern blotting. Basically, we're going to take a nylon membrane and we're going to transfer those fragments to that nylon membrane using capillary action. And what that then means is that we can then use either radioactive or fluorescent DNA probes, short DNA or RNA sequences, depends on which one we're looking at, and they're going to be complementary to our microsatellites. And because they're complementary to the microsatellites, they are going to form hydrogen bonds. And because they're forming hydrogen bonds, they are going to be able to, when we expose it to X-rays or UV light, they are going to make it visible so that these bands are able to be seen at the position that they're at. So the hybridization side of things is where you are going to use it in a way to visualize whether you've been successful. So as I said before, when we did it, so I'm not sure if it's the same what you did. So imagine you have got and again, I'm just making this up. This is not like the actual real thing, but imagine you've got a plasmid, and that plasmid is, I don't know, 1078 base pairs. And you have got another plasmid, and in that plasmid it's, I don't know, 948. And let's say we take a section of that. We want to take 85 base pairs. I mean, my scale is completely out, but never mind. Now, if we use restriction endonucleases and we use the same restriction endonucleases, what we would end up with is one fragment, which is 1000, let's say, and 70. No, that's too big. Let's say it's 1038. And we have another fragment, which is 40. So we get two fragments by using the restriction endonuclease on plasmid 1. On the second one, we end up with one that is 85 base pairs and another one which is, oh, Christine, why did you do this to yourself? My mental arithmetic when I'm tired is shocking. So I'm going to cheat and go to the calculator. What did I say it was? 948 minus 85. You guys can probably work it out faster than me. 863. I never trust myself mentally when it comes to maths. I always switch numbers around the wrong way. So I always double check. So yeah, so I've got another fragment, two fragments, which is 863 and 85. One that is 1038 38 and 40. Now, let's say if I change the color of this one to be red. So there's my 85, there's my 863. Let's just change that one. Oh, no, let's try that again. So this is now red. So what we've got then is we've got different colored fragments. And what I'm going to do is I'm going to try and stick them together. So if I've used the same restriction endonucleases, so I'll do here and here. So let's say I used ECO RI, and this one is Hindi 3. So I would have ECO RI here and Hindi 3. So what I've done is I've used the same restriction endonucleases, therefore they have the same recognition sites. So because they've got the same recognition sites, Skylar, I'm actually on, I don't know why it's not letting me do it, so I've had to go on my phone for TikTok. So I'm on my other channel, actually displaying it. I tried something different and then it wouldn't let me do it, so I had to just do what I'm doing now. I will try and get it fixed for next week. Not next week, for Wednesday. I'll try and fix it so it can do both. So yeah, so now what's going to happen then is if I go to the next, let's go to the next one. I have now got these different strands. So what could happen is I might end up with them recombining, and if they recombine and I end up with the recombination that I want, I would therefore get my 85 with my 1038. So when you add the two together, what are you getting? You are getting really bad. I should really be able to do this, but my brain is like, no, you're keeping too much in your head. 1123, or if it were to combine a different way, let's say it combined where I had the 863 combining with the 1038, then you would get a bigger strand, which would be 1901. So they would separate out in different ways. So you'd either end up with the single strands, the little fragments, the different separated strands, or you may end up with a plasmid where it's been recombined. And if it's been recombined in the right way, you therefore will get the size that you're looking for. So it's about knowing what it is that they want you to gauge from the information that they're giving you. So if I were to take my wells, I were to look at it and I were to get 1, 2 bands there, and then I was to get a band that's down here, I would know that this one is bigger than the original strand, and therefore that contains my recombinated plasmid, for example. But obviously you have a lot more to use as a way of confirming whether or not it has actually been transformed or not. Okay, so DNA profiling then can be used for forensic science. As I said, like if you are going to take a DNA sample, whether that's blood, whether that's semen, whether it's saliva, whether it's your root hair cells. So if you get a hairbrush and your hair has come out and it's still got part of the cells from the follicle there, they're going to be able to extract DNA from it. Skin cells. So any of these areas that have cells which have the DNA, and the DNA can then be extracted from, that can then be used for DNA profiling to figure out, do the bands match? If someone is heterozygous, so if you know the difference between whether you are heterozygous or whether you are homozygous for something, which is what we did. Remember when I said we tested to see whether or not we were heterozygous or whether we were checking our own cells. We were looking for microsatellites, and we were looking to see whether the individual was heterozygous or homozygous. So if they're homozygous, you would expect to see one band. If they were heterozygous, you would expect to see two bands. If someone was homozygous for, let's say, the longer number of repeats, they would have a band at the bottom end. If someone was heterozygous for the shorter, so let's say this one was 100, this one was 150. I'm just making this up. So therefore, individual A is homozygous for the 100. Individual B is heterozygous. They have one which has 150 repeats and one which has 100 repeats. And individual C is homozygous, so 2 of the 150 repeats. So this is how they can use DNA profiling to figure out whether or not someone was there, if they're a suspect, whether they're using the database. Oh, database, what does that bring in? That brings in our bioinformatics and computational biology. So this is where they bring lots of things together. Is this how they test for sickle cell traits? In my hospital work experience, the doctor said they test for 5 main genetic disorders when a baby is born. So I got genetic testing actually done for both my children before my children were born. And they are looking for, when they do the genetic testing, they are looking for the combination. So they can either do DNA sequencing, where they are looking at the actual sequence of the DNA bases, and therefore if they've got the DNA base sequence, they can then use that to figure out the likelihood of the trait being present. Or they can do it based on doing the likes of gel electrophoresis. So it depends on what test they're doing, but by removing a sample of the DNA, they can then use that to be able to figure out whether someone is carrying the allele for the trait or not. So someone who's got sickle cell anemia, they either have two so if they've got sickle cell anemia, it's a recessive trait, so they have 2 of the recessive alleles, where if someone is a carrier, they'll have 1 dominant, 1 recessive, and if someone is a wild type, so normal, they will not have the sickle cell trait. So that's down to potentially them looking at the proteins and the folding of the proteins, because the sickle cell hemoglobin is different to that of the regular hemoglobin. Somebody asked, Skylar, you asked, can you discuss STRs, short tandem repeats versus your variable number tandem repeats? So your tandem repeats are basically talking about whether you have 2 to 4 bases. So let's say it goes AT-AT, 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 or whether it goes G-A-T-T-A-G. So it's about these repeats. So when we're talking about the variable number tandem repeat, so you've got a set number of bases that can be anything between the 2 to 4, if it's the small ones, the micro ones, or variable number tandem repeats, you're looking at between, oh, see, I should know this, and off the top of my head it's completely gone, because it's 10 o'clock and it's my bedtime, my brain's gone, right, stop now. I want to say it's 15 to 1,000, hold on. Hold on. What is it? What is it? What is it? What is it? It is... There we go. Literally, my brain's shutting down. This is what happens when you get tired. So the micro ones, the short ones, are 2 to 4 bases. The mini satellites, the 2 to 40, but it's 50 to several hundred. Yeah, my brain did not remember that. Huntington's and Down syndrome, they can test. So yes, it's an interesting one. So Down syndrome is where you have trisomy 21. So Down syndrome is where they find a third chromosome at chromosome number 21. So you have 22 autosomes. Number 1 is the longest, all the way down to 22, which is the shortest. And then 23rd pair is your 6 chromosomes. So when you look at Down syndrome, then you are looking at the number of chromosomes at chromosome 21. However, oh, 1 second. The Huntington's, I'm not sure you can actually work that one out for when they're testing. So they test for things like, oh, there's one which is the cat calling one. It's such a weird name, but it means that when the baby is born, they have this like noise that they make that is like a screaming noise. It's really weird but interesting at the same time. Oh, that's good. That's now actually come up here. Interesting. Sorry, I was trying something new and now it has actually come up on here, which is really fascinating. So your personal statement mentions sickle cell traits, how it can be considered a defense mechanism for those. It's really cool actually when you look at sickle cell anemia, but it's not very cool for the people who are dealing with it, because obviously sickle cell anemia does protect you from malaria, which is why you find it in those places. However, when you have sickle cell anemia, you have to deal with the pain, the fact that it causes the blood vessels to get blocked in your joint areas. It's really not a very nice condition to have. One of my students last year, who lovely student, actually was in and out of hospital because of the flare-ups from their sickle cell trait that they had. Right, guys, my brain is shutting down. I can really feel it. I am so tiring. So tired now. I swear you can then use germline therapy. Well, hypothetically, obviously not. Yeah, germline is illegal. Well, so Huntington's is an interesting one because Huntington's is caused by a dominant allele. And so I'm not sure they can without actually sequencing it. I don't know. I need to look into that one. But they do genetic testing to find out if you are at risk of developing. And that's the key thing to note, that with any genetic disorder, whenever they do this, they are doing it and they're figuring out the risk factor of you getting it or of you passing it on. So someone who has, for example, cystic fibrosis, they may get genetic testing done to see if they are going to produce an offspring that has the disorder, because if they're a carrier and their partner's a carrier and they both pass on that recessive allele, that's going to cause problems. 3 sickle cell patients with crisis. Oh, and it made me want to work with them in the future. See, this is the thing, like when you figure things out, when you know more about it, when you see this for a real life situation, you then do want to try and find ways that you can help. And that's one of the things that I'm going to big myself up here, why I do what I do. I do what I do because I want to inspire those of you who are the next generation that are going to actually come up with these solutions, come up with these things that might help cure diseases that we have yet to figure out. Like, there's so many things out there that we just don't know. But with the advancements that we've got of technology, with AI, etc., we're learning so much quicker that there is so much more potential out there. And that's the key thing. But you need to understand it first to then be able to look at how you can help others. But guys, I am going to finish now because it's 5 past, so it is after my bedtime. And I will be back on Wednesday as always at 9 o'clock. So if you are interested, please do come and join me. Please, thank you for those of you who are sharing, liking, reposting my stuff, because that's really helpful. And if you bought my study guides, thank you so much, because that is what helps me be able to do what I love to do, and that is do things like this. Yeah, watch this space. I've got my patterns of inheritance one. I'm so excited. It's so close. It's so close to being finished. So that's the next one that's coming out. So yeah, guys, thank you so much for joining me, and have a good evening. Rest up. Go to bed.